You've swatted at a fly. You already know how that ends. Your hand moves. The fly is gone before it even registers as a threat to you. It just isn't there anymore. And you've probably told yourself the same story everyone tells. Flies just see the world in slow motion. Everything's fast for them. That explanation is incomplete in a way that's honestly a little embarrassing for us. Because a study published in 2026 just found that when a fly makes one of those split-second dodges, something happens inside its eye that shouldn't be physically possible. Not impressive, not surprising. Physically shouldn't happen, according to the fly's own hardware specs. Let's talk about the turbo boost hiding inside a housefly's eye. For decades, the standard explanation for "why can't I hit this fly" came down to one measurement: flicker fusion rate. Basically, how many separate images an eye can register per second before they blur into one continuous motion, like individual frames of a movie. For flies, that number sits around 230 per second. Ours is closer to 60. So the story became simple. Flies just have a faster frame rate. Except that number comes from testing a fly sitting completely still, looking at a steady flickering light. Calm conditions, a resting eye. Nobody had actually measured what happens inside that eye during the one moment that matters most. The middle of a full-speed evasive turn. The exact instant a hand is coming at its head. When researchers finally did measure that moment, the 230 number didn't just go up a little. It nearly quadrupled. Researchers at the University of Sheffield gave this mechanism a name almost nobody outside a neuroscience journal has heard yet. Synaptic high-frequency jumping. And here's the part that matters. It's not the fly's eye cells themselves suddenly speeding up. It's the synapse, the connection point between the light-sensing cell and the next neuron in line. That single junction shifts into a completely different gear. When it does, visual bandwidth jumps from that old 230 number to nearly 1000 Hz. Some of these synapses may be transmitting more information per second than any neuron ever recorded, around 4100 bits per second. To put that in perspective, that's not flies see fast. That's a single synapse potentially outperforming the fastest neural information transfer we've measured in any animal, anywhere. So the obvious next question is, how does a synapse just decide to get 4 times faster on demand? Here's the mechanism, and it's stranger than faster neurons. It's closer to what happens when you overdrive a guitar amp. Push a clean signal too hard into an amplifier, and instead of getting louder cleanly, the peaks get clipped flat. The smooth wave turns into something closer to a square wave. That clipping, that distortion, actually injects new high-frequency content into the signal that wasn't there before. That's essentially what's happening in the fly's eye. 6 separate photoreceptor cells pool their signals into one downstream synapse. That combined signal is clean enough and strong enough that it drives the synapse into a kind of clipping, squaring off the signal. And that distortion is exactly what generates the high-frequency boost. The distortion isn't the flaw here, the distortion is the feature. But, and this is the part that breaks the 6 is just better assumption, try this with just one photoreceptor feeding that same synapse alone, and you get nothing. Zero boost, just noise. It takes all 6 pulled together or the trick doesn't fire at all. Now, if you've seen any other coverage of turbovision claims before, there's a good chance it's centered on one detail: the fly's eye twitches. Tiny, constant micro-movements, even when the fly appears to be holding still. The obvious assumption is that the twitching causes the speed boost. That's the version that's going to dominate other channels' coverage of this. And it's wrong. When researchers built a model with a completely frozen, non-moving eye, no twitching at all, the high-frequency jump still happened. As long as that synaptic feedback loop stayed intact, the boost fired anyway. The twitching isn't the engine, it's a side effect. And here's the genuinely uncomfortable part, the part that fits this channel perfectly. That twitching isn't free, it actually costs to fly something. The timing noise it introduces reduces total information transfer by roughly 10% compared to a perfectly still eye. So the fly is voluntarily giving up speed. Why would evolution do that? Because in exchange, it gets something more valuable in the moment. Sharper spatial resolution. It's a trade, not a pure upgrade. A compromise. A little less raw data speed, a lot more detail. That's the overpowered but risky tax, hiding in an organ the size of a pinhead. Quick reality check. Because a fly isn't one animal. The housefly buzzing around your kitchen is not running the same hardware as a lab fruit fly. Housefly photoreceptors integrate signals 3 to 4 times faster, around 308 Hz of effective bandwidth versus about 72 Hz in a fruit fly, on the exact same test. Same basic eye design, wildly different performance. And it comes down to raw sensor density. Houseflies pack roughly 54,000 light sampling structures per photoreceptor. Fruit flies only about 30,000. The fly you can never seem to hit is, quite literally, playing a harder difficulty setting than the one in a genetics lab. Here's the detail that made me stop and reread the paper twice. When a fly starts an evasive maneuver, its actual physical response, the wing and body movement, begins within 13 to 20 milliseconds of the visual trigger. That sounds fast. It's not the strange part. The strange part is when that window falls relative to the fly's own eye activity. The fly's body starts moving before its own photoreceptor signal has even finished rising to its peak. It's reacting before its own eye has finished processing what it's reacting to, not after perception completes during it. And on top of that timing oddity, the resolution during that exact high-speed turn measures sharper than the physical anatomical limit of the fly's own lens, resolving details more than 4 times finer than its hardware should theoretically allow, at or below the literal diffraction limit of the eye. A system reacts before it's done seeing, resolving detail past its own lens's physical limit, during the fastest, highest stakes moment of its life. That's not fast. That's a nervous system that seems to be cutting corners. Nature shouldn't allow it to cut, and somehow it works. So next time you miss a swat, that's not just a fast bug. That's a nervous system running a real-time trade-off between speed and detail, pooling 6 weak signals into one distorted, overdriven pulse, reacting to a threat before it's even finished perceiving it. We spent decades measuring flies at rest and calling that the answer. The real answer was only visible in the half-second nobody thought to look at closely enough, the exact moment it mattered most. That's kind of the whole point of this channel. The most overpowered things in nature almost always come with a cost nobody advertises. If that shifts how you think about the next fly you can't catch, subscribe, and I'll see you in the next one.